The overall goal of the following experiment is to visualize mRNA expression patterns at the cellular level with high sensitivity and specificity. This is achieved by first taking formalin-fixed and paraffin-embedded tissue sections through a series of solutions to remove the wax, permeabilize the tissues, and block positively charged amino groups in the tissues that can yield a background signal. Subsequently, a DIG-labeled RNA probe specific for the gene of interest is infiltrated into the tissue sections, and the slides are hybridized overnight. Next, the slides are treated with RNase A to remove non-specifically bound probe from the sections, and then incubated with anti-DIG antibody that allows the visualization of the hybridized probe by a colorimetric chemical reaction. The results show dark blue signal detectable by light microscopy, specifically in those cells within the tissue in which the gene of interest is expressed.
The main advantage of this technique over other expression analysis, such as RT-PCR, microarray, or next-generation sequencing approaches, is that the in situ hybridization protocol shown here reveals the expression pattern of a gene of interest in its tissue context.
These methods include many steps that are best learned by visual demonstrations. We will show how to section and embed tissue, as well as key steps in the actual hybridization protocol that are difficult to master on your own.
The method of embedding paraformaldehyde-fixed tissues will vary depending on the type of tissue to be analysed. The most important point here is to ensure that the samples are oriented correctly for sectioning later. One method of embedding is to use plastic moulds and rings. To begin this procedure, warm up the moulds on a 60°C warming plate and then pour molten paraffin into the moulds. Next, use warmed forceps to transfer a tissue sample into each mould and orient it into the desired position. Carefully move the mould from the plate to the bench and place the white ring onto the mould. Add additional molten wax. Let the wax cool down and harden gradually. Prior to sectioning, warm the blocks to room temperature. Then trim each block into a trapezoid shape, leaving about 1 mm of wax around the sample. Place the trimmed block onto the microtome such that the longer of the two parallel faces is at the bottom. Carefully bring the block forward to the blade and make sure that the surface of the block is parallel to the blade. Section at a thickness of 8 to 10 microns. Align the wax ribbons on a non-sticky surface, such as aluminum foil, and select sections of interest under a dissection scope. Next, mark Probon Plus slides with a pencil. Do not use pens or markers because they will be erased during the in situ hybridization protocol. Distribute 1 ml of clean millicube water onto each slide. Carefully float sections of interest on the surface of the water at room temperature. Ensure that the shiny side faces the water. Transfer the slides slowly onto a slide warmer set at 35 to 37°C. This temperature range, as opposed to the commonly used 42°C, prevents the formation of bubbles under the sections. After 5 minutes, pour off the water carefully but in one smooth movement, so that the ribbon is lowered down onto the slide. Let the slides dry for at least several hours or overnight at 37°C so that the tissue adheres. To prepare tissue sections for in situ hybridization, they are first deparaffinized and rehydrated. For deparaffinization, incubate the slides in 2 changes of Histoclear solution for 10 minutes each. To rehydrate the tissue sections, pass the slides through a series of decreasing concentrations of ethanol for 30 seconds each. During the protease treatment, which is not shown in this video, prepare a glass dish with 0.1 molar triethanolamine solution and set this up on a stir plate in a fume hood. Immediately before placing the metal slide rack into the solution, add 1.25 ml of acetic anhydride into the triethanolamine buffer and stir well. In addition, set up the clamping system and stand for holding the slide rack. Reduce the speed of the stirrer. Clamp the slide rack onto the stand and lower the rack into the solution, keeping it elevated above the stir bar. Continue to stir slowly for 10 minutes. After being rinsed in PBS and dehydrated again through an ethanol series, the tissue sections are ready for hybridization. Place the slides on a clean surface and let them air dry completely for 5 to 10 minutes. Prepare the probe hybridization buffer mixture by adding the denatured probe to 80 microliters of hybridization buffer for each slide. Mix well by pipetting, but avoid making bubbles. Pipette the probe hybridization buffer mix onto the right edge of the slide. Gradually, lower a cover slip onto the slide, making sure that the hybridization solution covers all sections without any bubbles. Tap the cover slip lightly with your finger where bubbles form to displace them from all tissue sections. Do not pull the cover slip off, as this will damage the sections. Place the slides in a humidity chamber containing Whatman paper moistened with Millicue water and largely covered with parafilm. Seal the box tightly. Incubate the slides at the desired hybridization temperature, typically 50 to 55 degrees Celsius, for 16 to 20 hours. At the completion of the hybridization protocol, carefully remove the cover slips from the slides. The cover slip may simply fall off when the slide is placed upright, but if not, do not pull off the cover slip, as this will damage the sections. Instead, immerse the slide in warm 0.2 times SSC for 1 or 2 minutes to wash the cover slip off. After removing cover slips, place the slides in a rack and wash several times in 0.2 times SSC at 55 degrees Celsius. After RNase A treatment, transfer the slides from the rack onto the bottom of a flat plastic box and cover with a minimal volume of blocking solution. Block the slides for 45 minutes at room temperature on a slowly shaking platform. Next, apply a minimal volume of antibody solution directly onto each slide. Incubate the slides in the dark for 2 to 3 hours at room temperature. When the incubation is complete, transfer the slides to a clean, flat box and wash the slides 4 times with a minimal volume of washing buffer on a shaking platform at room temperature. Rinse the slides once in TBS and twice in TN buffer. To apply freshly prepared staining solution to the slides, first pour the staining solution into a small plastic weighing dish. Then, sandwich 2 slides together with the sections facing each other. Dip the sandwich into the solution, allowing capillary action to pull up the solution. Drain the slides on a Kim wipe. Fill the slides with staining solution again. The slides may need to be tapped as the solution flows in to avoid bubbles. Place the slides in a plastic box and store at room temperature in the dark. Representative results obtained with different probes on Arabidopsis tissues are shown here. The purple signal provides a direct visualization at cellular resolution of the expression pattern of the gene of interest. Panel A shows the localization of shoot meristemless 1 transcripts in the vegetative shoot apex. Note the presence of purple-blue signal in the indeterminate cells of the meristem and the lack of signal in the surrounding leaves. Panels B to D are sections of Arabidopsis torpedo stage embryos, where B shows clavata3 expression in the few embryonic stem cells, C shows ATML1 expression in the epidermal layer, and D shows the lack of signal in sections probed with a random negative control RNA. These next images show results obtained with different probes on maize tissues. Panel E shows the localization of the STM homolog knotted 1 in the developing maize embryo. Note the strong signal specifically in the embryonic meristem and root. Longitudinal sections through the vegetative shoot apex show that ARF3A is expressed on the abaxial bottom leaf surface in panel F, and the ATML1 homolog OCL4 is expressed specifically in the epidermis in panel G. As expected, no hybridization signal was observed for the negative control probe in panel H. The expected results of the different checkpoints during the in vitro transcription of probes are shown here. The in situ hybridization probes are checked on a standard agarose gel after in vitro transcription, after DNase treatment and subsequent purification of the in vitro transcription reaction, after carbonate hydrolysis, and when ready for use. For probes over 250 base pairs in length, such as probe 1, the carbonate hydrolysis will yield a range of smaller probe fragments. This figure shows the results of colorimetric qualification of probes by dot blot analysis. Dilutions ranging from 10 to the -1 to 10 to the -5 of a 100 nanogram per microliter control probe and of 3 newly labelled DIG labelled probes are spotted on a transfer membrane, incubated with anti-DIG antibody, and assayed. The analysis suggests an estimated concentration of 100 nanograms per microliter for probe 2, 10 nanograms per microliter for probe 3, and 1 nanogram per microliter for probe 4, indicating that probe 4 is unlikely to yield a good in situ hybridization signal. Finally, these longitudinal sections through the vegetative shoot apex from maize provide a comparison between a non-specific background signal and a well-working probe. Panel A shows a non-specific background signal, while Panel B shows a specific in situ hybridization signal for the OCL5 probe in the outer cell layer.
After watching this video, you should have a good sense of how to perform many of the tricky steps in the in situ hybridization protocol, such that you can determine the precise spatiotemporal expression patterns of your favorite genes.